Hello friends, this is the class on gluconeogenesis. This process actually helps in production of the glucose, and this is an universal process, and you can find this process in animal, plant, fungi and microorganisms. So for that reason it is called a universal process of glucose production. Now in animal cell there is no enzyme present that can produce pyruvate, which is a key component of this gluconeogenesis and works as a substrate from which the glucose is produced. This pyruvate, which is a 3 carbon substrate, is this one cannot be produced from the 2 carbon substrate like the acetyl CoA. So during the fat metabolism in the beta oxidation, lots of acetyl CoA are produced, but it cannot really contribute into this gluconeogenesis pathway. Whereas in plant there is one cycle present which is called a glyoxylate cycle, which produces succinate by joining two acetyl CoA molecule together, and it subsequently produces oxaloacetate that can contribute as a substrate in the gluconeogenesis pathway. So all those precursors of the gluconeogenesis pathway are the lactate, pyruvate, TCA cycle intermediates and glucogenic amino acid. So now let's go to the pathway. This is the pathway. Before I really start, I would like to show you the complete pathway. This is the complete pathway, and I must say that gluconeogenesis is actually an anti or opposite directional pathway of the glycolysis. In glycolysis you produce the glucose produces pyruvate. Glucose is a 6 carbon substrate. It produces pyruvate of 3 carbon product of 2 molecule. So here the red arrows, downwards red arrows is indicating the glycolysis pathway, and the upward blue arrows are indicating the gluconeogenesis pathway. Here it is not just opposite to glycolysis, as there are some crucial steps. One of them is this one, where this pyruvate cannot really directly produce phosphoenol pyruvate. It has a different kind of pathway where oxaloacetate is the intermediates, is a substrate within this pathway. Whereas during glycolysis, this phosphoenol pyruvate produces pyruvate by the enzyme pyruvate kinase, and one molecule of ATP is produced. So this pathway, I would like to give details of this portion of this pathway in this slide. Here it is the pyruvate molecule of 3 carbon. Let me change the color of this. So this is a 3 carbon molecule, and look at this oxaloacetate. It is a 4 carbon molecule. How it happens? This pyruvate produces oxaloacetate by the enzyme called pyruvate carboxylase. From the name of this enzyme, you can understand that one carbon dioxide molecule, one carbon, has been added in the form of carbon dioxide, and ATP is also present over there during this mechanism. And look at the structure where this carboxyl group has been added. Here is the methyl group, and one hydrogen has been removed, and into that empty hand of the carbon this carboxyl group has been added, and here is the 1 2 3 4. This is the carbon number of the carbon chain, and so the oxaloacetate is a 4 carbon molecule. Now this oxaloacetate has to come into the cytosol. From this mechanism happens in the mitochondria. So this oxaloacetate has to come into the cytosol, and it is not just an easy task because oxaloacetate cannot penetrate the membrane of the mitochondria. So during the production of the phosphoenol pyruvate by the enzyme called phosphoenol pyruvate carboxylase kinase, if this enzyme, this PEPCK, phosphoenol pyruvate carboxylase kinase, if it is present in the mitochondria, then there is no problem. The enzyme with the help of GTP, it takes out one carbon dioxide and produces phosphoenolpyruvate, which is of 3 carbon. And then the subsequent steps of the glycolysis takes place, and subsequent steps of the gluconeogenesis occurs, and the glucose is produced. But if this enzyme phosphoenolpyruvate carboxylase kinase, this enzyme is present in the cytosol, then there is a problem, because this oxaloacetate cannot penetrate the membrane of the mitochondria. So here what thing comes into play, that is, this malate is produced. Malate is the reduced form of the oxaloacetate. Look at this. This is the carbonyl group, and here this is a carbon with the hydroxyl group. So this carbon has been reduced, and for this reaction the enzyme malate dehydrogenase is responsible, and this NADH plus H plus works as a cofactor which donates the hydrogen and reduces oxaloacetate into the malate. So this is not NADH, this is NAD plus. The NADH plus H plus actually donates the hydrogen and get oxidized and form the NAD plus, whereas this hydrogen is taken by this carbonyl group, and it gets reduced and produces hydroxyl group. This carbonyl is now reduced into hydroxyl group, and the oxaloacetate produces malate. Now this malate, this molecule can pass through the mitochondrial membrane, and then again it get reoxidized. This is a wrong side of the arrow. The arrowhead should be in this side. So here again this NAD plus comes into play, and it takes away the hydrogen from the malate, and it reoxidizes this malate in oxaloacetate, and now this NAD plus takes the hydrogen from the malate and becomes NADH plus and H plus, and goes into the electron transport chain in the mitochondria to produce ATP. And now this oxaloacetate, with the help of the phosphoenolpyruvate carboxylase kinase present in the cytosol, this is PEPCK enzyme present in the cytosol, now produces phosphoenolpyruvate. This reaction takes place in the cytosol, and we had a problem that how we will pass this oxaloacetate from the mitochondrial membrane to the cytosol. This is the process. That oxaloacetate that reduced into malate then cross this membrane of the mitochondria and then reoxidize to produce oxaloacetate. Then with the enzyme present on the cytosol, that is phosphoenolpyruvate carboxylase kinase, produces phosphoenolpyruvate. Now this phosphoenolpyruvate then again goes straight into the opposite direction of the glycolysis, that is the general gluconeogenesis pathway. It produces 2-phosphoglycerate, then 3-phosphoglycerate, and so on and so forth, and then produces glucose. So this portion, I guess it is clear to you. And now let's move into the next slide, which is this one. This pyruvate produces phosphoenolpyruvate by through this pathway where the oxaloacetate work as an intermediate, and then 2-phosphoglycerate is produced by the help of this enzyme called enolase. And as I have already discussed the details of the glycolysis in my previous video, so I guess you understand the function of the enzyme according to the name of the enzyme. And then 3-phosphoglycerate is produced by the enzyme called phosphoglycerate mutase. This mutase enzyme, this word mutase means that the enzyme has a role where it actually it moves a group from one carbon atom to another carbon atom into a carbon chain. So this phosphate group, which was in the second carbon, now it is in the third carbon, and this reaction has taken place by the help of this enzyme phosphoglycerate mutase. Now this 3-phosphoglycerate produces it is not 1,2, it is 1,3 this is 1,3-biphosphoglycerate. From that, glyceraldehyde 3-phosphate is produced, and this glyceraldehyde 3-phosphate now produces dihydroxyacetone phosphate. From the dihydroxyacetone phosphate, 1,3-biphosphate is produced, and then this step, again, it's a critical step. This in case of this glycolysis, fructose 6-phosphate with the help of 1 ATP produces fructose 1,6-biphosphate by the enzyme called phosphofructokinase, and this is a rate limiting step in the glycolysis. Likewise, in case of gluconeogenesis, this step is also very crucial, and it is governed by the enzyme called fructose 1,6-biphosphatase. Here there was 2 carbon at the first and the 6th carbon position. Sorry, 2 phosphate was there at the first and the 6th carbon position. Now this ATP has been hydrolyzed from the first carbon position, and this fructose 6-phosphate, the phosphate is left only at the 6th carbon position. And this biphosphatase, these enzymes are responsible for hydrolyzing ATP into ADP and PI by with the presence of a molecule of water. 1 molecule of water should be added here, and here here the phosphate group actually is removed in the form of this inorganic phosphate. And the next step is, from the fructose 6-phosphate, glucose 6-phosphate is produced by the phosphohexose isomerase. From the name of the enzyme, you can understand that this fructose and the glucose are the isomers. So when I am changing a structure from one isomeric form to another one, where this fructose is changed into the glucose, so the enzyme phosphohexose isomerase comes into play. And the last one is the glucose 6-phosphate, which produces glucose by the help of this glucose 6-phosphatase. Here it is another important step where one phosphate group, which was in the 6th carbon position, is hydrolyzed by the presence of a molecule of water, and this phosphate group is now removed in the form of an inorganic phosphate, and this glucose is produced. So there are 3 important steps. One is this one, where the oxaloacetate is works as a key, as an intermediate between these steps of pyruvate phosphoenol pyruvate production from the pyruvate, and this is the second step, which is this one. It is the second step where fructose 1,6-biphosphate produces fructose 6-phosphate by the enzyme fructose 1,6-biphosphatase. And this is the third step, which is a very crucial step, in which glucose 6-phosphate produces glucose by the help of this enzyme called glucose 6-phosphatase. Now all these enzymes have some inhibitor. This fructose 6-phosphatase is inhibited by, I'm just writing down this name. This is fructose 6-phosphatase, which is inhibited by fructose 2,6-biphosphate. This is one of the molecule that actually inhibits this fructose 1,6-biphosphatase, and then fructose 6-phosphate and AMP is there, which works as an inhibitor. Now there are some more molecules which actually activates this enzyme. Those are citrate and ATP. So these are the activators. Alright, so I guess this was fine, and I guess this video will be useful for you to understand this gluconeogenesis pathway. And now I will be uploading my next videos, but if you have liked this video, you can you can subscribe my channel in the YouTube. This is the link. And for more videos you can visit my website, which is easyscience.weebly.com. And I will also want your feedback about these videos, whether it was helpful to you, and what are those videos that you want me to produce for you, so that you can understand this science easily and a better way. Thank you for listening to my video.